Hi guys, Dr. Gillard here once again. This is Tuesday's lecture. That's tomorrow, but we're getting this done ahead of time because I got that big 2-hour CVPP tomorrow I got to do still. But anyway, this is week 1, Tuesday's endocrinology lecture. Remember, endocrinology is part of GIGU, Spring-2020. Here we go. We left off on starting to talk about the angiotensin-aldosterone system, or the R2A, Renin-angiotensin-aldosterone system is where we're going. A little review just to kind of get our brains warmed up on this. We will be having, I guess right now it's not too important, but remember this thing. What are we looking at here? Robert, what are we looking at? You paying attention, Robert? This is the distal convoluted tubule, right? This is the loop of Henle. Remember all this stuff? This is the nephron. So the star of the show, we're going to talk about the juxtaglomerular apparatus here in a second. We've got Bowman's capsule in here. And yeah, I think you know all this stuff. The proximal convoluted tubule, loop of Henle, the thick limb we'll talk about a little later on, distal convoluted tubule. The very end part, Guyton talks about this being called the connecting tubule. A lot of principal cells live here, so that's an important piece. Then, of course, we have the collecting duct here. All right, the other thing weird, I think this is the last little review here. The other thing weird about the kidney is it's got 2 sets of capillary beds. I mean, there's no other place in the body I know that has 2 capillary beds supplying one organ. So this is the one we're familiar with. These are the glomerular capillaries. You definitely know the afferent arteriole and the efferent arteriole. Another weird system, right? Since when does an arteriole go into a capillary and back into an arteriole? This should be a venule, right? But that's not what happens. So this is this glomerular capillary is weird. And then it goes down here into the peritubular capillary, which is what you're used to. There's an arteriole here, the end of the efferent arteriole, and there's another. Then we become venous. There's a venule right here, and things are back to normal. But yeah, pretty weird. Remember the stuff that the juice that is squirted out here from the blood is called the filtrate. Filtrate goes through the nephrons, and a lot of the water and the sodium and such are returned back to the blood. The interstitium, of course, it has to flow through the interstitium into these capillary beds. Talked about the interstitium a little bit last time. All right, so let's talk about this R2A system. A whole bunch of AKAs. I like to call it the R2A. Some people call it the Renin-angiotensin system, the RAA system, the R2AS system, the RAS system. There's a lot of AKAs, so who knows which one will show up on boards. I will tell you one thing about boards. None of those are those abbreviations. In boards, you have to spell it out. So it's going to be the Renin, one of these, the Renin-angiotensin system, or the Renin-angiotensin-aldosterone system. All right, so this is it's the most important controller of the sodium concentration in the body. And therefore the total body sodium is controlled by the R2A system. And via its regulation of sodium, it controls these other primary functions. So if you control sodium, you control blood pressure. If you control sodium, you control blood volume. All right? And we'll get into all how this stuff works, and you know how a lot of this stuff. I'm just hitting this because of aldosterone, right? That's a hormone. That's my domain. Yeah, and there's a bunch of them, right? ADH and stuff. Therefore, it also controls sodium homeostasis. It's a super busy system, right? That darn Angiotensin II that comes out of this. We'll look at that, how it's born in a second. But, oh my goodness, the kidney's probably the star of the show. It's kind of interesting because renin is actually born from the kidney, and it comes back to roost as angiotensin II, which has big effects on the kidney. Central nervous system is affected. The heart and lungs are deleteriously affected. What's that mean, deleteriously? Adversely. What's that mean, adversely? Evilly. It's damaging. Angiotensin II and aldosterone people with heart disease, chronic hypotension. We'll get into that when that comes. But people with any decreased heart output always have the R2A system turned on to correct that hypotension. Therefore, they always have too much aldosterone and always have too much angiotensin II around, and that can wreck the heart. All right, so adrenal glands, it hits the adrenal glands as well, right? ACTH hits them. Smooth muscle, it can flat out contract the arterioles, not as quickly as sympathetics can, but it can contract those arterioles and venules. So a good way to understand the R2A system is to think about somebody who has a bleed, and I mean, this is ridiculous here, but what if someone has an aneurysm that's popped in a small artery and they're bleeding, they're leaking blood, and they're not enough to really even know it. So that's a good way to study this. The R2A system, if you got something like this, the R2A system is not going to save you. It's the sympathetic tiger, I call it, and we'll look at that. But it's kind of a slower system, but it's definitely better at restoring the volume. You're going to lose blood volume. That's going to need to be replaced by water. So it's key for restoring that. And it can stop bleeding as well, but just not as quickly as the sympathetic system can. How does it bolster blood volume? How does R2A system get blood volume and therefore blood pressure up after it's dropped? Well, in general, we're going to talk about all these systems in detail, but let's just go through them quickly. Good, review board question here. Notice all the stars. Oh, I'm getting pretty excited about this question, especially this slide, because there's 3. I could say, which one of the following? All of the following bolster blood volume, lost blood volume via the R2A system, except blank. So, yep, ADH, check, that does it. Aldosterone, check, it does it. Thirst centers, check, it does it. And I could say something weird that would be, but devious though, right? Weird but devious, enough to trick some of you. So anyway, let's go through these. Antidiuretic hormone, of course, causes the kidney to conserve free water. Sodium is not conserved. I'm going to say that a million times, and some of you are still going to miss that, but don't miss that. So if you conserve water, that bolsters the blood volume, which increases the blood pressure. That's the goal, right? We got somebody bleeding, someone with low pressure. It also triggers the release of aldosterone, right? And we'll look at that mechanism. Aldosterone conserves water too, but it conserves salt as well, so that's good. Same thing, if you increase salt and water, you increase the blood volume, that increases the blood pressure. It also goes to the hypothalamus, the thirst centers of the brain, and makes the patient very thirsty, or makes the person very thirsty, right? That's always a bad sign. If you pull up in a car accident and someone's hurt really bad, you don't see any visible blood, but they're super thirsty, probably means they're bleeding internally, and that thirst mechanism has been turned on. Number 4, it causes vasoconstriction. Don't forget this. People think only the sympathetic system causes vasoconstriction of the arterioles and the venules, but that's not true. We'll learn angiotensin II can directly bind to the smooth muscle and cause contraction. So very powerful mechanism of smooth muscle contraction. It's just a little slow is the problem with it. It also stimulates the adrenal medulla to release epinephrine or adrenaline. Those are, you got to know the difference. A little bit of norepinephrine, or I shouldn't say a little bit. Mostly epinephrine is released. Norepinephrine is not as much, but there's still a significant amount. What's the other one? Which one did I miss? Good, somebody got it. Dopamine. Dopamine is the other one that's released. Just a little bit of dopamine. Okay, and then the epinephrine selectively shuts off systems by vasoconstricting arterioles we don't need. For example, the skin. Someone who's bleeding internally, they start to get a little shocky looking, don't they? Their skin gets cold and clammy. How come? Epinephrine is cranking out and it's shutting off the skin to conserve blood for the brain and things that need it more. It makes the heart pump harder. It binds to the SA node, and I think we'll look at that more when the time comes. But now that's not going to bolster your blood volume any, but it's going to bolster your blood pressure because it's pumping the remaining blood around quicker and trying to make up for that decreased volume. Now let's back up and look at the R2A system a little more closely. The best place to start the discussion of the R2A system is at the kind of the birth of renin. That's the start of the R2A system. That's why it's called the renin. That's why it gets the R first. So the kidney, we have to start there. So the very first thing that happens is the arterioles that are around the nephron. They sense a decrease in blood pressure or decrease in blood volume. Those two go hand in hand. And that sets off a chain reaction. So let's talk about it stimulates in general. Be careful, the juxtaglomerular apparatus, okay, that contains two players, right? Let's check it out. So the kidney has 2.5 million little blood pressure sensing devices in each nephron. Who are they? Juxtaglomerular apparatus. So the juxtaglomerular cells, that's not an AKA for the juxtaglomerular apparatus. These are cells that live in the walls of the tunica, or live in they replace the tunica media. They live in the afferent arterioles. So they live within the tunica media of the afferent arterioles. Only in the arterioles that are near the glomerulus. The other part of the juxtaglomerular apparatus is the macula densa cells. Okay, these guys sense pressure. These guys sense what? What are they tasting the filtrate for? Who knows that one? Good, somebody got it. Sodium content. They don't care about pressure, they care about the sodium concentration of the filtrate. Together, these guys make up the juxtaglomerular apparatus. Getting pretty excited here, lots of stars popping up. Okay, let's take a look at this, what we talked about. Here's a glomerulus, a little different location than we saw it before. Here's a small picture of it. There's the loop of Henle there in this little cartoon. There's the proximal convoluted tubule sucking out sodium at the beginning. And there's the distal convoluted tubule. The very proximal portion of the distal convoluted tubule is where these things live. Let's blow it up and look at them. So here's the afferent arteriole. There's the glomerular capillaries. There's the efferent arteriole. And we got these weird little tunica media cells, and those are the juxtaglomerular cells of the juxtaglomerular apparatus. All right, so those guys are sensing the pressure that's flowing through here and the volume of blood that's flowing through here. These guys are going to get stimulated if the pressure decreases, right? And what do they release? Oh, a bunch. You got that one. Renin. Renin is dumped right into the interstitium here, and it jumps back into the bloodstream, and it's off to the races, as we'll see in a second. Okay, so that's the juxtaglomerular cells. So, like everything I said, they're baroreceptors, constantly monitoring the pressure of the blood in that afferent arteriole. Upon sensing any decrease in pressure, they get upset, and we won't go through the mechanism, but they release renin into the bloodstream. Renin is an enzyme. It's a protease, and it's a cutter. It cuts, and it cuts its substrate. We'll look at its substrate, angiotensin, Angiotensinogen here in a minute. So the release of renin from the juxtaglomerular cells is also the rate-limiting step of the R2A system. What in the world does that mean? The rate-limiting step. That means that this is Angiotensin II, which is the workhorse of the R2A system, can only be released via the release of renin. So this is the rate limit. If we injected extra renin into the body, more R2 or more angiotensin II would be released, because the ACE enzymes can handle way more conversion of angiotensin I to angiotensin II. But the bottleneck of this system is the production of renin right there. Good board question. What does renin do? It's a protease, so it's always floating around a little bit, the bloodstream, looking sometimes very low levels, but it's looking for its substrate, and its substrate is angiotensinogen. That's its mate, so to speak. When these two hook up, renin cleaves the tail off angiotensinogen, cleaves off its tail, and it creates a new molecule, and that's called angiotensin I. Okay, angiotensin I is just a cleaved, shortened version of angiotensinogen. I used to get a little biochemistry, but I took that out because we got so much stuff to do. Angiotensinogen is a liver protein. This is the one that is made, make sure you know this word, constitutively. This one is secreted from the liver, angiotensinogen, all the time. And so it's always floating around. Right? It's not stored at all. It's got to be constitutively secreted. It's always secreted. And it's the major binding molecule for renin. It's considered a prohormone. It can also be converted into other angiotensins. There's angiotensin IV that I'm not going to talk too much about, but it can be converted directly into angiotensin II. Not very much, but for board purposes, it's converted into angiotensin I. Angiotensin I has no biological activity, so its only function is to float around the body and look for its mate. And its mate, who it wants to bind with or hook up with, it is somebody named angiotensin-converting enzyme, ACE. There's a ACE1 and ACE2. Is this ringing a bell? Anybody been looking in PubMed at COVID-19 and what's the pathophysiology of that? Anybody done that yet? Somebody? Yeah, guess where COVID-19 enters the lung tissue? ACE. That's its target. COVID-19 binds to ACE2. And I've got a kind of exciting paper that just came out. I'll show you here in a second. So ACE is an enzyme which sits on the endothelial cells along with other things like the receptors that grab cholesterol and things like that. The endothelial cells are quite busy, but especially in the lungs, and it's in other places too, not just the lungs, but in the lungs, they're really heavy with these ACE enzymes that sit on the endothelial cells. Okay, so what does ACE do? ACE is very powerful. It converts angiotensin I into the sometimes evil and incredibly busy angiotensin II. Who's that little guy? You guys, anybody still watch cartoons? I don't see my grandkids watching this anymore. That's the Tasmanian devil. It's probably politically incorrect, but he was part of the Bugs Bunny series. But for your purposes, that is the angiotensin II. This guy is everywhere. If you ever saw this cartoon, he's like a tornado, and he just flies up and down. He digs in the dirt and pops back up the dirt and goes through trees, and he's just crazy. And that's angiotensin II. This thing is all over the body. It can do some good if you're bleeding. It can cause some trouble, though, and it does. It can wreck your heart if you don't take meds to calm it down. So angiotensin II is the biologically active form of all the angiotensins, the major one. There other ones have some activity. We won't worry about those. Super diverse molecule. It's all over the body. Half-life is really short. It's less than 60 seconds, so it's not around very long. Now, here's some brand new slides. I actually put these in about half an hour ago as I saw this paper pop up. So, as I said, COVID-19, that's that virus that's causing trouble all across the world. That guy uses ACE, specifically ACE2, to gain entry into the epithelium of the lung tissue. It can get in other places. It can make your joints ache and your muscles ache because ACE is otherwhere, but ACE is really heavy in the lung tissue. And so it gets inside the epithelial cells and inside the parenchyma of the lungs, your body's immune system says, holy S, who is this invader that's gotten into our lungs and attacks? And now we have a wicked inflammation because so much of it gets in. The inflammation doesn't know any better to back off, and it's really the person's own immune system, the inflammation and the pus and the exudate and just the fluid that's generated drowns the poor person who's affected of this stuff. And it shuts down their respiratory system to the point they need a ventilator. You go on a ventilator, you got anywhere from a 50-50 chance of coming out of it. I've seen some studies say you got about a 30% chance coming out of it. The problem is not the breathing. While you're on the ventilator, those darn bugs overflow the body's inflammatory response, and they get into the bloodstream, cause septicemia. Super dangerous, we'll talk about it, super common way to die. So it's that darn ACE is where they come in. So who cares about that? Whose grandma and grandpa is on ACE inhibitors? Have you ever heard of those? So here's a new study came out last month, March 23rd. And sure enough, I always suspected this. So ACE inhibitors and angiotensin receptor blockers may increase the risk of severe, fatal COVID-19 manifestations. And it makes perfect sense because people with failing hearts, their heart can't pump enough blood, so your blood pressure is always low. And who's turned on if the blood pressure is always low? The R2A system. And who's the king, who's the busybody, or who's the main player of the R2A system? Angiotensin II. So angiotensin II is being oversecreted, and it damages the heart, and we'll look at that more effectively. So to prevent your heart from being damaged, you need to block the production of angiotensin II by either taking inhibitors, which competitively interfere with the binding to ACE, or you can let the angiotensin II be formed, and you can block the receptor sites for it so it can't bind. But the body, just like with people with GERD who take PPIs, proton pump inhibitors, same deal. The body sees that ACE is not doing its job, so it grows new receptors, and that's the problem. So people on these medications have way too many ACE enzymes and too many ACE receptors, and therefore COVID-19 has a field day getting into the lungs. There's so many places it can get in, and that's at least the theory. This is the first one that I haven't dug into it too deeply, but that sure makes a lot of sense to me. So if I was on ACE inhibitors, I wouldn't be on them anymore after I read this article. I would wait till we got a vaccine for it. And then I could start them later when this is under control, because that makes a lot of sense to me. Anyway, kind of digressed a little bit. So there's angiotensin. It's on the loose. All right, so let's talk about AT2, some of the AKAs there. Amongst other things, it is the most potent vasoconstrictor out there. Watch this word, pressor agent. I saw that in Guyton. Pressor agent. What the heck is a pressor agent? Pressor agent. It increases blood, and a pressor agent is something that increases blood pressure. Pressor, like vasopressin, right? That increases blood pressure as well. So it's a very strong pressor agent, and it can bind, as I said before, we'll come back to it officially, but it can flat out bind to the tunica media and cause a contraction of that tunica media and arterioles and increase your blood pressure that way. There is an angiotensin III that is also created by ACE, has similar effects to angiotensin II, triggers the release of aldosterone, as just like angiotensin II. We're kind of getting ahead there. It's made from angiotensin I. It's a little bit different. It's not made by ACE. That was my bad there. It's made from angiotensin I. It's converted right into ACE3 by a aminopeptide ACE A. 1 star. Am I probably going to ask this question? Probably not. It's probably not going to be asked. There's even more. If we really want to go crazy, I mean, if you want to see what angiotensin I can become, ACE2 makes angiotensin II. There's up to 9 angiotensins. It gets really complicated, so there's no stars on this. But this is just showing you that there's a lot more to the story. But what I'm teaching you is plenty deep for even the toughest board question. They would never go this deep. Okay, so we talked about the juxtaglomerular cells. What about the other member of the juxtaglomerular apparatus? That's the macula densa cells. Let's talk about them. They live in the proximal portion of the distal convoluted tubule. Kind of showed you a picture of them. Super close, kind of they can elbow the juxtaglomerular cells if they want to. So really close to them. They're also a member of the juxtaglomerular apparatus. Don't miss those easy board type questions. Remember, the juxtaglomerular apparatus is not an AKA for the juxtaglomerular cells. This is like the major category here. The two categories underneath are macula densa cells and juxtaglomerular cells. These guys are not, they do not sense pressure. These guys are chemoreceptors. They sense chemicals, or AKA for chemoreceptors is osmoreceptors. So they're not pressure sensors. They're not baroreceptors like the juxtaglomerular cells are. We'll look at them again. So if we go through, there's the proximal convoluted tubule, loop of Henle, distal convoluted tubule. So there they live, right in this, would be right here in that piece. And if you blow it up, there's what they look like. This time the endothelial cells are actually, they're modified endothelial cells. Remember, these juxtaglomerular cells, the endothelial cells are normal. These are smooth muscle. Replace some of the smooth muscle here. But notice they're touching each other. So they have the ability to communicate. Now, what do? So I always remember this snake. So they're constantly sticking their little snake tongues. Do I have my type? Oh, yeah, I did. So if they have like these little snake tongues, and they're constantly tasting the filtrate moving through. And they're tasting it for the concentration of sodium. It has to be just salty enough for them. It can't be too salty, or it can't be especially can't be under salty. They freak out if it's under salty. So they're constantly tasting the filtrate for the sodium ion concentration. Chloride as well. They're tasting for chloride. Sodium, chloride go together. Sodium is the one most talked about, though. So what about hypotension? How does this work? Well, if you have hypotension, the flow through the proximal convoluted tubule, there's not going to be much pressure pushing it, right? So the flow through here is going to be, I guess I don't have it drawn here, but it's going to be really slow. So what? How's the flow of the filtrate going to be if you have hypotension? Is the filtrate pushing through here going to be fast? No, it's going to be slow, right? If the pressure is slow, not enough fluid drains in, and it's just everything's slowed down because there's not much pressure. So who cares about that? Well, now we need to talk about the proximal portion of the proximal convoluted tubule. A lot of sodium. There's cells and channels that suck out sodium here. So I use the analogy of a potato picker. Here's a conveyor belt. So the conveyor belt's going, and we have workers on each side of the belt looking for the bad potatoes. And if they see them, they pick it out. The belt has a certain speed. They can't pick out maybe every single bad potato because it's going too fast, but they try their best. What happens if in the bad potatoes, let's substitute bad potato for sodium. What happens if we slow this belt down? Like way down for somebody who has hypotension. There's not going to be much flow through here. Those potato pickers are going to have a field day. They can pick every single sodium molecule out of here. And that's exactly what happens. It makes the filtrate really decreased in sodium. And so who cares about that? The potato pickers don't care. But we know somebody does. The macula densa just absolutely freaks out. Oh my God, there's not enough sodium in the filtrate. Holy ass, we got to do something about it. So decreased sodium and chloride too sets it off. They elbow their buddies here and say, hey, we got an emergency. There's decreased sodium in the filtrate. We got to fix that. We need more pressure because this isn't right. And there's a complicated mechanism we won't talk about, but they communicate. So here's the question, the test question I ask if people screw up. So do the macula densa cells directly release renin into the bloodstream? No, they indirectly release renin because they don't have the ability to make renin. But they elbow their buddies here who can make renin, and they'll go, okay, we'll make more renin. These guys are already making renin because the pressure is already slow here. It's low. So they're already freaked out making renin, but now the macula densa is freaked out and makes them release even more renin. So that's the story of the juxtaglomerular apparatus there. What about the response? Everything we said, everything I just said. Yep. So we don't need to talk about that again. But just remember, the renin is ultimately converted into angiotensin II, and that's the little monster that gets us in trouble. So we're going to look more closely. I think we're going to do that next time. Look more closely at how this affects the body, and we'll do that. I don't know if we'll do that the next lecture, or I have to see how the schedule. I think it's all, because it's not on Friday anymore. I think it's all kind of smashed together on Thursday. So we'll see where we go. I'll take a look at the schedule, but we may probably just do 2 of these lectures a week, and the rest will be GI/GU stuff. All right. Thanks for watching. We will see you soon because I got to do the CVPP 2-hour lecture next. Okay. Take care.